When a T-cell encounters a target antigen-presenting cell, it forms a specialized contact site called the immunological synapse. The lymphocyte centrosome translocates to the synapse in order to polarize the microtubule cytoskeleton and direct the secretion of cytokines or cytotoxic agents toward the target cell. Fanni Gergely from the University of Cambridge and Cancer Research UK is interested in how T-cells rapidly polarize their centrosomes during an immune response.
The current model was that you somehow tether dynein and dynactin, the microtubule minus N-directed motors, to the cell cortex just beneath the immunological synapse, and these dynein and dynactin then cross-link microtubules that come from the centrosome, and then they would pull the centrosome towards the immunological synapse. And then it docks in the plasma membrane. So we were asking the question whether can we find new kinases that are potentially involved in positioning the centrosome in these cells. We tried to screen a small panel of kinase inhibitors, and the one which gave us the absolute best result was Casein kinase 1 inhibitor.
Gergely and her colleagues, Deborah Zyss and Hani Ebrahimi, found that Casein kinase 1, or CK1 inhibitors, reduced T-cells' ability to polarize their centrosomes toward the immunological synapse. These inhibitors block the delta and epsilon isoforms of CK1, both of which localize to the centrosome. The researchers knocked down both isoforms by RNAi and found that CK1 delta was required for centrosome polarization in T-cells.
If a centrosome is not polarized, it might be because it didn't translocate, or it could be that it just was not maintained, in this docked position. And the real-time experiments told us that actually it was the translocation that was affected by Casein kinase 1 delta depletion.
Control T-cells rapidly polarize their centrosomes to the immunological synapse, but centrosome translocation failed or was delayed in cells lacking CK1 delta. Centrosomes successfully docked with the immunological synapse once they reached it, however, and Zyss et al. Found that other aspects of the synapse were also unaffected by CK1 delta depletion. Synapse assembly appeared to be normal, as indicated by the appropriate accumulation of integrins, actin fibers, and the T-cell receptor at the site of cell-cell contact.
Signaling at the synapse was intact, and the recruitment of dynein dynactin to the immunological synapse was unperturbed, suggesting that our effect probably occurs downstream of the recruitment of dynein to this site within the cell.
CK1 delta bound to microtubules and localized to dynamic filaments protruding from the centrosome of activated T-cells. Moreover, the researchers identified a short sequence motif in the C-termini of both CK1 delta and CK1 epsilon that mediates protein interactions with the microtubule plus N tracking proteins EB1 and EB3.
Therefore, we tested for binding, and indeed we confirmed that these proteins could actually bind both EB1 and EB3. But it turned out that this motif is neither sufficient nor required for binding.
Instead, CK1 delta's kinase domain was required for the interaction with EB1, and for the protein's association with another plus N tracking protein, the dynactin subunit P150 glued. The researchers also found that CK1 delta phosphorylates EB1 on a specific serine residue. But although the kinase's SQIP motif wasn't required for this phosphorylation in vitro, these amino acids were essential for CK1 delta's function in T-cells. Mutants lacking the SQIP motif couldn't restore centrosome polarization to cells missing the wild-type kinase.
So we believe that while this is not necessarily what mediated binding to EB1 in vitro in a test tube, somehow in vivo, this very short 4 amino acid motif has got a much more important role to play, and in the future we hope to figure out why.
Because CK1 delta interacts with the plus N binding proteins EB1, EB3 and P150 glued, Zyss et al. Decided to investigate the kinase's effects on microtubule behavior.
And what we found is that indeed Casein kinase 1 delta in lymphocytes is really important to maintain microtubule growth speeds. So when we deplete or inhibit Casein kinase 1 delta, the mean growth speeds of microtubules are reduced. T-cell
microtubules don't need to be dynamic in order to polarize the centrosome. However, the decrease in microtubule growth upon CK1 delta depletion may limit the number of microtubules long enough to be captured by dynein at the immunological synapse. Indeed, inhibiting microtubule growth with the drug coulcimide also reduced the ability of T cells to polarize their centrosomes.
When we actually assayed T cells that are not activated and T cells that are activated via conjugation to an antigen-presenting cell, we actually observed a reproducible increase in microtubule growth rates in the activated T cells. So the model what we put forward is that Casein kinase 1 delta, together with either EB1 or P150, are important to increase the microtubule growth speed upon the activation of T cells. What is the signal that transmits this from the T cell receptors to Casein kinase 1 delta? We don't know. But we think that Casein kinase 1 delta potentially exerts this effect near the centrosome to generate the long and stable microtubules that are probably important for centrosome translocation via the dynein-dynactin complex at the cortex.
Alternatively, Gergely says, CK1 delta may help EB1 and P150 glue to anchor microtubules to the centrosome, allowing dynein and dynactin to haul the centrosome toward the immunological synapse.
And I think the other really interesting question is what exactly phosphorylation of EB1 by CK1 delta achieves. Because this phosphorylation maps to a linker region within EB1, and we don't really know the function of that particular region.
In the meantime, you can read more about how casein kinase 1 delta promotes the translocation of a T cell's centrosome to the immunological synapse in the paper by Zyss et al., published in the November 28, 2011 issue of the Journal of Cell Biology.